advise on this project with a number of others, and I'm sure we universally would like to congratulate you and colleagues on reinventing Irish haematology, shall we say, and very impressed with what we see so far. So, as Michael indicated, I've been flapping about in clinical trials for a number of years and thought I'd like to convey a few thoughts about how we've been thinking about optimizing what is a relatively rare patient population. AML is not a common disease, and it's not been a big issue till about maybe 10 years ago because there wasn't that much available for new treatments. But of course, we now have a lot of potential treatments, and the issue becomes one of how can you look at these new treatments efficiently and reasonably accurately. And this, I think, apart from other things like biomarkers and companion diagnostics and molecular characterizations and prognostic groups, really needs revamping of clinical trial design. So we've gone through this sort of process in the last few years when I was on the AML group. Now, the AML group in the UK was funded by a rather obscure grant going to Oxford, which allowed us to do what we liked from the MRC. And this started in 5 centres in the 60s. And there has always been a clinical trial available since the 60s for AML in the UK. Initially 5 centres, and this is built up so that we're now in the second generation of haematologists where this was a standard of care, being in the clinical trial. So we're very fortunate in that sense. Originally it was based around Oxford, and Richard Peto, of course, Sir Richard Peto, was instrumental, particularly interested in paediatric leukemia, but he was, of course, a dominant force in statistics. And interestingly, they never believed that there was any point in doing phase 2 studies or early phase studies. They wanted big numbers in the trials for statistical relevance. And I was beginning to get uneasy about what I call the Petoization of AML trials, because if you cannot see a difference in an outcome within a relatively moderate number of patients, then it's probably not going to be clinically that meaningful. So he used to like 2,000 patients per arm, you see. So you can see that this rather tied up our numbers. So you can see from this slide that the recruitment of centres in the UK and the number of patients entering the trial per annum has risen over the years. This is slightly out of date, but we probably get 65% of all the patients in the country in our various trials. It usually comes to about 12 or 1,300 patients per annum, which is the biggest trial group in the world, actually, in AML. And you can see here, oh, that's that one, yeah, I'm not paying attention. And this allows us to ask a number of questions. You can see that over this period up till 2012, this was 17,000 patients in the last 20 odd years. The important thing about trials is not the number of patients, but the number of randomizations you can achieve. So that's 33,000 randomizations were achieved. And we asked 45 questions. Now, this was done led by Keith Wheatley statistically, who had moved to Birmingham, who was a bit more of a free thinker. But they were very keen on factorial design. In other words, you would enter an initial randomization and then there could be another randomization at a later point. The danger of that is that if you get something happening in the first randomization that affects how patients respond to the treatment, the second randomization, you have an interaction and you can't interpret it. So the statisticians know how to interpret or look for an interaction between treatments, and actually it turned out to be quite rare in AML. So we were able to ask all these questions. And the other thing is that this became a standard of care, so the district haematologists were involved. And this became political. Is it right for AML to get treated in a district hospital? Should they go to the transplant centres or the teaching centres? So we looked at that. And you can see here that there actually is no difference in survival achieved by the big centres or the small centres. So this was good for the small centres, but also good for the patients. They didn't have to travel. And I think it kept the standard of haematological management up in the district hospitals. This is younger patients. If you look at the older patients by big centres or small centres, the big centres do worse. And this is probably because they're prepared to take on more difficult patients who are referred in, but we don't really know that. So anyway, this strategy of traditional trials, large trials, did bear some fruit, as you can see on the left-hand side. This is paediatricians, this is paediatric ALL, and you can see that treatment has improved substantially in children. They probably have an expectation of cure of about 70%, 75%. And then on the right-hand side, even the young adults under 60, are improving, such that you can look at a patient under 60 and think you've got a 50-50 chance of getting away with this. We know, and I'm not going into detail on this, that the individual patient's risk differs substantially. The strongest indicator, but not the only indicator, is cytogenetics. Age, secondary disease, presenting white count, how they respond to their first course of chemotherapy are also very important. And we use these factors in a weighted index to decide who should go to transplant. Why did these, we didn't see any differences in most of these 47 randomizations, so why did improvement take place? And it's probably because supportive care was better. You can see here over time that the 30-day and 60-day mortality has dropped. So this is nursing and supportive care, line access, blood products, etc. But also deaths in remission obviously dropped. So supportive care is probably the main component of why the treatment got better. Some subgroups, and this looks at the two favourable subgroups on the left panel here. On the left panel here is acute promyelocytic leukemia. You can see that we expect now to cure about 90% of these patients. And that is now done without chemotherapy at all. This is combining arsenic and atra, retinoic acid derivative. And this was not invented in the molecular lab. It's known how these agents work molecularly. But this was done in China by people just putting the drug in a test tube and seeing how the cells reacted, and that was given to patients. And you can see the effect. So that's a remarkable biological consequence of just empiricism, really. And on the right-hand side, you can see the core binding factor leukemias who've obviously improved, and actually the outcome's about 80% for them now. So there are subsets where it's pretty clear we're doing well, but these are distinctly the minority. Here is the real challenge. On the left-hand panel, you've got our history of 20 or 30 years with patients with bad chromosomes, and you can see that we've made no difference to these patients. Even although they respond initially, they will relapse very quickly. And on the right-hand panel is the patients over 60. Now, these are patients over 60 who were thought to likely to be suitable for intensive therapy. It did not include the unfit patients. These were patients who got intensive therapy, and you can see again we've made very little progress here. So these are the areas where our trials have failed. And I remember being challenged by a district hematologist saying, why do you give us treatment here that isn't suitable for most of our patients and doesn't work for the ones for whom it is so suitable? Now, in this disease, why these differences occur is partly explained by an aggregation of adverse prognostic factors that occurs in the older patients. So how should you proceed to try and get a new treatment identified quickly for these? And these are the majority of the patients with the disease. Should you do phase 2 trials, unrandomized, just continue to look, as some designs do, or should you do it some other way? Now, we've been wedded and continue to be wedded to randomization in this disease. And the reason is a lesson taught to me by Keith Wheatley, the statistician, who identified this treatment called SAB And you can see here that this was statistically better than the daunorubicin thiaguanine that we used at that time. And so we sent this letter in as SAB looks better. What is SAB? Same as before. This was taken, this shows you how the basic control arm moves with time in this disease. So things get better, partly due to the supportive care, and therefore you can easily mislead yourself if you look at a phase 2 trial. And this is, of course, what's happening when you do a phase 2, pharma thinks they've got the cure to leukemia, it goes into randomization and it doesn't beat or it barely beats the opposition. Here's another example, again looking at just low-dose RSC in 2 periods of time in the elderly unfit, and you can see that the remission rate originally was 8%, and recently in a control arm of another trial it's 19%. So things change with time. So randomization, in our view, is essential when you're assessing a new drug seriously. So of these 40-odd randomizations, we can only claim that the treatment improved the outcome in about 4 or 5 situations. I've mentioned APL. The immunoconjugate, gemtuzumab ozogamycin, which is not approved in Europe, is only approved in Japan, so we buy it from Japan because we showed that it did improve survival by a little bit. And in these good prognosis patients, it improved it a lot. Low-dose RSC giving to old patients who were originally just given supportive care and not much else is better than giving just supportive care. I'll show you the data. It's not a great treatment, but it's better than supportive care. And I think what's going to be important is that minimal residual disease or measurable residual disease, whether by multi-parameter flow or by RQ-PCR or possibly next-generation sequencing, is going to be very important to define the prognosis of individual patients. Not just showing the average outcome as a prognostic group will show, but showing an individual patient. And I think this will probably define whether they move into a response measurement rather than looking down the microscope alone. So although the outcome is improving, it really isn't primarily due to the treatment. This is the most recent trial in young patients, and these just give you an idea of the numbers that entered each randomization. I think it's difficult to start getting smart in younger patients where the outcome is gradually improving, but these interventions were trying to hone the optimize the chemotherapy, and there's still an argument to do this sort of trial. The second bottom one is quite interesting. We're interested to know whether having minimal residual disease monitoring is of benefit to a patient. You know, we can do this, it is prognostic, but is it predictive? Does it tell you what to do? Does it make a difference to survival? So we're randomizing patients to be monitored or not monitored. It's the only study of this type. And it's a sort of public health question. Does the technology, which is difficult and expensive, does it improve the survival of patients if you have this data available to you as a clinician? So that will be kind of unveiled probably later this year or next year. So can we go to smaller groups of randomized patients? I asked the question, you know, we've got 1,000 patients in each arm, and I used to say to the statistician, look, if you don't see anything at 1,000 patients in each arm, what about looking at 500 or 250 in each arm? Is it likely to be different? So when you subdivide by cohorts of 50, how likely are you to get the wrong answer by reducing the numbers? It seemed to me the CTSU, the statistical argument was to be sure that a treatment doesn't work. If you need 1,000 patients in each arm to show that, I think we kind of lose interest as clinicians. And here, this looks at a series of patients, and you can see that, in fact, there's not any occasion where it's clearly going to give you a different answer. This was a trial that overall was negative between the two arms, and you can see that each of the sequential cohorts of 50 were more or less inconclusive. So it's not that risky to reduce the numbers in this disease. So the problem is that we've randomized a lot of patients. In the elderly in particular, we've not improved the outcome. Any benefit is small, and if you're introducing a new drug and you only see a small benefit, you've got an economic issue about whether this is really affordable. And also, of course, there are all these new drugs, and how can we incorporate them into the treatment of AML without, falling behind, as it were? So we came up with this design called Pick a Winner. In retrospect, we've looked at about 10 or 12 options, and this is effectively a randomized phase 2, but it's more complicated than that. There is a trial design called multi-arm, multi-stage design, whereby patients are put on an experimental arm, and then you pick the one that looks best and you carry on with that one. It's not quite the same as that. So what we do in the Pick a Winner is that the patient, and we've focused on the elderly, so-called unfit, first-line treatment, they come in and they get randomized to whichever option in the trial they're eligible for. So we may have 5 new drugs and a control arm. And they come in and they can get, if they're eligible for all of the arms, they get randomized to one of the treatments or the control. So immediately you see if they can get into any of them, your control is common. So you're not, you should have about 1 control to 4 experimental treatments. So you save on patient numbers in that respect. If they're not eligible because of cardiac or some other exclusion, then they only go into the arms for which they are eligible. And a key issue with pharma who participate in this is that you are not comparing experimental drug A with experimental drug B. You're only comparing the new drug with the control arm in each case. And that reassured pharma, and we've never really had a problem of pharma being concerned about their new drug being compared with that new drug. Of course, you get a pretty good idea which one's better, theoretically. So then you can have a look at whether you're likely to achieve your aims by having an interim futility analysis after a certain number of events. And that is an important milestone at which you can jettison treatments that are not going to fulfill your ambitions. What your ambitions are is really up to you. In our case, we wanted to double the response rate as an indicator that we might improve survival. So you want to see that at that stage at least there's some improvement going on, and then you can drop treatments that are not working, and they can enter, rather than 4, they can enter 3 or 2. If they pass that first futility hurdle, then you're in a traditional phase 3 trial with overall survival as the endpoint. So this isn't Pick the Winner designed by Daniel Sargent, where that looks at trials only individual treatments and there is no contemporaneous control in operation. It just looks at 4 potential new treatments and picks the one that looks better. That doesn't necessarily tell you it's better than your standard of care. And it's really a refinement of the multi-arm, multi-stage schedule. So it comes as more efficient. But it's more than that, because we include in the protocol, in the patient information, drug X. And drug X is the ability to bring in a new treatment by protocol amendment. So you're not writing a new trial, you just do an amendment. Now, this was an issue both with the MHRA in the UK and with the National Ethical Committee. And I went to see them without an actual proposal, just to go over this design. And the Ethical Committee got very concerned about it until I sort of challenged them and say, well, what are the Ethical Committees? They said, no, this is a separate trial. And I says, well, what's different ethically about randomising a patient there and randomising a patient here, and they couldn't answer, but they slipped out, well, we don't look at amendments just as closely as we look at new trials. So I said, okay, well, you know, look at our amendments as closely as you look at a new trial. So we compromised. And the MHRA were a bit more open-minded to it, but were concerned that any amendment might modify the original aims of the trial. So if you do this sort of thing, you have to, I think, be prepared to take on the regulatory thing. And now that I work in pharma a wee bit, we are going to take this sort of idea to the FDA and EMA to see if they will buy into that sort of design. So you're randomising between a control arm and a novel treatment arm. You have the ability to sequentially introduce new treatments as you go along, drug X, as it were. And it's important, though, that you don't use the control on day one and compare it with drug X that happened 2 years later. They have to be contemporaneous randomizations for the reasons that I mentioned about how the control arm can change without apparently any other, for no apparent reason. So the issue is, what is your endpoint? Now, to make this really count, you want to get an endpoint that's clinically meaningful. It's a surrogate for survival. But you want that to be a frequent event, and you want it to be an early event. So an elderly AML death fulfills these criteria, or remission as the endpoint. So we chose complete remission as our initial endpoint. So you would know that endpoint in a patient in 2 or 3 months' time. So you're not waiting for overall survival down the line. You can make your decision based on that sort of event. But it had to be, we also wanted that endpoint aspiration of improving the endpoint, which is about 15% in low-dose RSC, for example. We wanted that to achieve a 30% CR rate. So these were the criteria that we would apply to assessing in the futility analysis whether a treatment was worth pursuing. And then the Data Monitoring Committee would have to have a charter that incorporated, you know, what the general rules were. So dealing with our endpoint, this was a randomised trial which was published nearly 10 years ago, and it was the first randomised trial in the so-called elderly unfit. In AML, in these days, a lot of patients didn't get any treatment. They were given supportive care. They might have been given drug like hydroxyurea to control their count, but the aim was quality of life and keeping them out of hospital, keeping them free of infection, giving them blood products. Definitive treatment was not thought to be suitable. So we had to define a standard of care, and we chose low-dose RSC. This was not popular in the UK. The reputation was all this does is make patients thrombocytopenic, doesn't do anything. But this is one of the 4 studies that the DMC stopped because it was, the study arm was better. It doesn't look great, but it was better. But the key thing here was that the patient who benefited were the patients who entered remission. Patients who didn't enter remission didn't benefit. And if they got into remission, which was about 20% here, then their remission lasted 15 to 18 months. So it was kind of worth having. So we went forward on the basis that CR was this early endpoint that we had to deliver improvement on, and that would translate into a survival benefit. That was the assumption at the beginning of the program. So we wanted to double the CR rate. We would plan to evaluate after 50 patients were treated on the arm, and then another one, if necessary, at 100 patients treated on the arm, to give the DMC a chance to say, look, boys, this is not, you know, you may have improved the remission rate, that's fine, carry on. But a later futility would say, well, you've improved the remission rate, boys, but it's not actually translating into survival. So there are various ways of turning off the investigation and focusing on the other drugs that are in the program. There are other ways of calculating the difference that is worth getting, and you could say, well, we'll look at the NICE criteria for funding of a drug eventually. So you can see here on this table, if your drug's going to cost 40,000 quid, on the left-hand panel, the 1-year survival has to be increased by 89%. This is what the threshold of the NICE sort of means. So you could actually power your study on the basis if you happen to know what the drug was going to cost. But the drugs we were working with were not approved drugs, of course, so we didn't know the cost. So the danger is that if you apply criteria and you set the bar too high, or too low, then you either accumulate treatments that are worthless or you eliminate treatments that would be beneficial. So Robert Hills, who is our statistician, and I may say one of the best decisions I made in my career was to appoint him to the department as a senior lecturer in translation statistics. So he simulated something like 10,000 trials to get a probability of when you should look and what you should see if it was a doubling of remission rate. And so he worked out all this. To this day, I don't understand it. But he worked all this out, and indeed, his rules apply to the decision that the DMC makes. So the essential elements are randomization between one control arm and several novel treatments. You've got to set your threshold, and you know, you could do this in myeloma, you could do this in CLL, you could do it in certain solid tumours, provided you've got a reliable endpoint that happens frequently and happens relatively early, then it's useful. So you need a meaningful surrogate, in other words. And then I think the big thing for me was you're not writing a new trial every year, you can just introduce drug X. And as long as everybody knows what you're doing, the regulators, the ethical committee, and the patient, then this appears to be acceptable. So what we're doing here is saving patient accrual that is unnecessary by early closure. Excuse the vocabulary of flogging a dead horse. But simultaneous flexible randomization means that you're only using one control. And also, if you've got a trial where you've got if you've passed the first hurdle and you're going on to survival and you want a number of events out here, and once you get to these numbers to do your assessment, you would have a whole lot of patients at various stages, but you could actually close down that arm and let these patients mature to the endpoint. And these other patients can go into the other arms of the trial. So you're not wasting patients' time or wasting patient resource in that sense. So does it actually work? We've been running this in the UK for about 10 years now. We've looked at 10 new treatments. How many of them got chopped? 5 got culled. So if we had done trials, and there was always a good reason to look at this drug, we would have done 5 individual trials, hundreds and hundreds of patients probably, and come to a negative conclusion. 4 of the 9 passed the first hurdle. In other words, the CR rate looked to the DMC to be potentially going to double the response rate, and there's 1 under consideration. Here are the actual treatments, the GOs, the gemtuzumab ozogamycin, the immunoconjugate, arsenic trioxide, tipifarnib, the sapacytabine, vosaroxin, the combo, the quizartinib, the AC220, ganetespib, the HSP90 inhibitor, all look great drugs in the lab, by the way. And there was some initial evidence in phase 1 that they did things. And then tusidostat, which actually is our company's aminopeptidase inhibitor. And you can see that some of these with the green ticks did double the remission rate and carried on. Others with the red cross fell at the first fence. They didn't improve the remission rate. However, these 2 at the beginning, although they doubled the remission rate, they did not improve the survival. So I thought, well, that must be because you're bringing higher risk patients into remission and they'll relapse earlier. Well, that wasn't the case, actually. The difference was that the patients who failed initial therapy didn't do so well than compared with low-dose Ara-C. And if a patient did well and got in remission and then failed, they didn't survive as long as if they were in remission with low-dose Ara-C and then failed. So the reasons were a wee bit complicated. But you can see that even though these two did produce benefits in remission, they didn't improve survival significantly. The Ganatespib did jump the first fence, carried on, but the company bailed out because their small cell carcinoma trial failed. So that was stopped, but when it was stopped, there was no difference in survival. And the others are ongoing. And there are a couple of drug X's introduced to the program. So it was clear that maybe remission as an endpoint is not that clever, because although it doubled, we weren't able to produce survival. But also there are new drugs like the demethylation agents who promote stability of disease without getting in remission. So what do you do, and they're licensed in Europe. So what do you do about that? Well, one of the options is to make your control into doctor's choice. So the doctor chooses what they would do. I think that's a bit messy, but at the moment, neither demethylation agent is approved in the UK, so this is viable at the present time. And low-dose Ara-C has never been shown to be inferior in survival terms statistically than the demethylation agents. So one of the issues is, would a program like this complicate matters for the hematologists? Would it be confusing for the clinician, for the patients? And in fact, the actual recruitment in this age range in AML went up by 500%. So this sort of activity is not a problem for patients. Patients are quite keen to get new treatment. And so there is an unmet psychological need. So why have we failed? Well, we've obviously not got drugs to deal with difficult situations in AML. But have we failed? If you double the remission rate, but don't improve the survival, is that a failure? It seems to me, if you're in your 8th decade, that the quality of what you've got left may be just as important as the quantity of time you've got left. And hematologists, although you do the EORTC, quality of life stuff, we've not been good at emphasizing the benefits of being in remission for the time you've got left, even although the time you've got left is not longer. So now we build in as much quality of life stuff as we can as well. And, you know, quality of life does weigh in the minds of the regulatory bodies as well. So we conclude that CR is not always the reliable endpoint. It depends on the mechanism of the drug. So I think this is somewhat repetitive. There's a definite inefficiency. I mean, I've been at the ASH meeting, spoken to a company, and introduced the drug in April by protocol amendment. So you know, you're not doing a 2-year development program to get a new treatment in. And from the company's point of view, this is great. I mean, timelines are very important to pharma. And also we have a QP release pharmacists. So all they have to do is give us the drug, and our pharmacy will sign it off that it complies with manufacturing. They'll put the signature on the form, and that will do for European use, that it's manufactured to European standards. So it can be done very quickly. And if you know your ethical committee and the MHRA and they know about it, they can handle this fairly promptly. One of the issues, I think, is making sure your sponsor's happy. And I remember when the European directive came out, Cardiff University had the grant for a big MRC trial, and I went down to see them and said, now, this trial requires a sponsor. The MRC is not sponsoring it. That's, yes, what do we do? I said, just sign here. And they signed, and they were the sponsor. They hadn't a clue what they were doing. But actually, they learned a lot about sponsoring trials and are happy to sponsor in France, New Zealand, Denmark, Australia. We would have gone into Italy. So, you know, if a sponsor really understands it, they regard it almost as a badge of honour as well. And that's terribly useful. So, I think this could be, I can see situations in other hematological diseases where you could do a similar design to trawl through these new treatments. Now that I've got into pharma, I'm kind of I was trying to set this up in pharma. There are some reservations, recruitment. Sometimes the investigators get confused if they only have rare patients. If you get too many options in your trial, it will take you a long time to get to your endpoint. So you want to balance the recruitment you get against the number of options. We think 4 is about right at any one time. That should give us the answer in 12 to 15 months, given our usual rate of recruitment. Clearly, there's an issue with the wrong surrogate endpoint. That's something that people have to be aware of, and we learned the lesson the hard way, I think. But still, you're wanting an endpoint as early as you can reasonably get it for efficiency. And then the DMC used to be controlled right June and October or whatever, really has to be assembled as an event-driven date. In other words, when you get your events, get the DMC organised. One of the problems in the elderly AML, of course, is should your patient get intensive therapy or not? And if you're over 70, then who says you won't benefit from intensive therapy? So this is a controversial area when it comes to talking to the FDA. They'll say, well, why didn't this patient get your conventional intensive therapy? And we're not very good at deciding that, but we're much more cautious in providing reasons about why these patients didn't get intensive therapy, and that helps them orientate to the point. Well, what happens if one of your study arms becomes the winner? Because this won't be a licensed drug. So you can either ignore that fact and just carry on and say, very good, we've got a winner, take that away and try and get it licensed. Or you can go to doctor's choice. For example, if we had azacitidine in there, for example, I think the investigators could have got hold of that, and they could get hold of that, and that would become a doctor's choice type of thing. So that's difficult, but as you can see, we've not had to face that challenge yet. What happens if there's a subgroup that clearly benefits from the DMC, whether it's a molecular subgroup? Well, in theory, when you carry on to the next stage, you could limit the entry criteria at that point. Now, that's effectively introducing another drug X, because you would have to do you'd have to analyse these separately, but it's doable. It can work. So it's popular. It can be delivered in a multi-centre setting, and actually many of these drugs were equivalent as being in a 1B situation. That they hadn't got any further than that, and they're developed in many situations. DMC should be event-driven, I've said. You have to be careful about your endpoint. And then the issue, I think, which is a more recent thing, is defining that being in remission is a good idea in AML. And survival is not necessarily the total. So this is repeating some of this stuff. The issue of companion diagnostic comes up. We think it's very important to store material at the beginning for whatever technology by whoever can look at it and see if there is an enrichment signature that could be useful in the future. Having gone into pharma, as I said, I tried to interest pharma in this, and it's clearly easier if the pharmaceutical company owns the drugs you're looking at, but it's not impossible that various companies could join a program like this. By and large, you have to know the dose you want to use. So you're in phase 1B territory, And then you could, from the beginning, you could randomize all the patients. You could look at the randomized patients on study and look at their safety as your safety signal. You keep randomizing, and then you look at patients on the study arm, not against the control arm, for an efficacy signal, perhaps as a simultaneous 2. If it passes these hurdles, that means you've got all these patients counting towards your phase 3. You're not starting a new trial. But we've not succeeded in doing that just yet. So, as usual, I've gone on a bit long, so I'll thank you for your attention.
How quickly do you have to get out of here? So thank you very much, Alan. We've some time for some questions, if there's any questions from the audience. Peter?
Thanks very much, Alan, and thank you very much. And I just want to applaud you for the marathon effort, because AML is a very challenging disease to treat, as we all know, as hematologists, and your efforts are really important. Can I ask you, do you see any particular drugs that catch your imagination, or that you think are going to significantly change the landscape going forward? Because as a clinician, we've been trying to get involved in the MRC trials and struggling to do that, but also we're looking for hope for our patients, and overall, as you say, we're relying on statisticians to identify minor changes, whereas what we're hoping for is a drug that's really going to be very easy to see as clinicians, and that would make a big difference. Do you see any hope on the horizon?
Well, if I was still in the game, I'd be very interested in the BCL2 inhibitor. I think that's very encouraging at the moment. The IDH1, IDH2 inhibitors are interesting, but logistically extremely difficult to know how you would do a phase 3 trial in that situation. It's not common. 50% of them never relapse in the first place, so it's even less common if you're doing it in relapse refractory. One of the new drugs, of course, that surprisingly has shown benefit in relapse is the Celator drug, the liposomal daunorubicin Ara-C combo. Now, the interesting thing about that's mixed in a ratio, as you know, doubles the survival at 2 years. Very surprising result. But what it might tell us is that if you can fix ratios of combinations and fix them in a liposome, that may be a new strategy for treatment. And anyone who's done combo experiments in the lab, looking for synergy, will know that the ratio of each drug can vary, the response can vary enormously. But I think we need to pay more attention to immunological control, because this is going to get very difficult in a rare disease with multiple subsets. And I think the immunological issues are important as well. We can have a generic treatment. So, I mean, that's what I would be interested in if I was still interested, had to be interested.
Okay, well, just before you go, Alan, I heard a rumour somehow that it might be actually your birthday today. You certainly didn't deny it, and so Alan has to leave us because he's going back to Scotland for a birthday celebration. So we have a small little gift for you here. So Sandra will explain. But just something as a little token of appreciation for your efforts in coming here on your birthday. So thanks very much, Alan.